that can observe evolution on ecological timescales, with the classic example being Presention. And understanding how contemporary evolution works is really important for getting a better understanding on, say, evolution of antibiotic resistance, or understanding how organisms respond to changing environments. The problem is that studying contemporary evolution requires genotype, phenotype, and fitness information across many generations, and so very few systems have these kind of data. To date, most population genetic studies tend to try to infer information about past processes using current day patterns, and even if we have multiple time samples from a given population, it's often pretty sparse. And so ideally what we want when we're interested in looking at short-term evolution is a system where you have complete pedigrees through time, complete fitness and genotype information for every individual in the population through time. Of course, such a system doesn't really exist, but if you have data similar to this, you can learn a lot about the role of short-term selection in maintaining genetic variation. So what I'm going to talk to you today is a system where we have some data and we're starting to look at, we're using different ways to study evolution in the past like 10 years or so. So I work on the Florida scrub jay. It's a cooperative breeder that's federally threatened. This bird is non-migratory, highly territorial and philopatric, and so what that means is that individuals born in a given area tend to stay there, so we can follow the same individuals throughout their lifetime. They're socially and mostly genetically monogamous, and so we can construct accurate pedigrees from field observations alone. And the nice thing, the reason I work on the species is that there's one population at Archibald Biological Station that has been intensively monitored since 1969. And there's been a substantial field effort, and so every month the entire population is census. So we have accurate lifespans for every individual in the population. Every nest in every territory is found and monitored, which means we have complete life histories for all of our individuals, and we can actually measure annual and lifetime reproductive success for all of the individuals in our population through time. Every individual, so all nestlings and immigrants that show up in our population each year, is caught and banded. We have a blood sample taken for them, so we have this archive of blood samples for DNA. Every territory every year is mapped, and we also have a whole suite of ecological data. So we have information on habitat composition, weather, food availability, fire history, etc. And so what this has resulted in is this collection of individuals for which we have life history and morphological data, and this deep pedigree going back in time. Our full pedigree actually looks something like this. And so this makes the system uniquely suitable for asking questions in evolutionary genetics. So during my PhD, what I did was build genomic resources for the species. So we discovered genome-like SNPs, sequence and assembled the genome, sequence and assembled the transcriptome to help annotate the genome, and we're currently working on building a lineage map. So today I'm just going to focus on the SNPs. We use genotyping by sequencing in a panel of individuals to do the SNP discovery and then design custom Illumina B chips for the genotyping, which is really nice because we have really high quality genotypes using this B chip and don't really need to worry about genotype probabilities as much. So the data set, after a lot of QA and QC, we're left with about 12,000 SNPs across the genome in about 3,800 individuals. So here I'm showing you the number of adult breeders in our population and the number of babies born in our population through time. The gray bars show the total number of individuals, and the blue bars show the number of individuals we've genotyped. And so you can see that we've almost completely genotyped the entire population in the past decade. And so the first thing we wanted to ask with these data are, do we see significant changes in allele frequencies through time in our population? And so for the most part, you would think, okay, we'll just do Wright-Fisher simulations and see if we get larger allele frequencies than expected by drift. But one thing to keep in mind is that the population pedigree actually structures the way that drift can occur in the population. And so if we can consider the pedigree, we will have more power to detect selection in our population. And so what we did is to do gene dropping simulations. So the way this works is that you start off you have the pedigree of your population. You start off by assigning genotypes to the founders, so the original population. Here we're going to start with the birds in 1990. And then you simply simulate Mendelian transmission of the alleles and the pedigree. So we wrote software to do these simulations. You run them a million times. And at the end, you get the expected distribution of allele frequencies in the population under neutral processes. So here, for example, I'm showing you one SNP in the gray, showing you the expected allele frequencies for the population in 2013, given our starting allele frequency of like, I think the starting allele frequency was about 0.57 in 1990. We do the simulations on the pedigree, generate what we expect to see under drift alone. And in most of our SNPs, you can then compare the observed frequency to expected. And if the observed frequency is within the 95% confidence interval, it's a neutral SNP. We do, however, see about 67 SNPs where the observed allele frequency shift is much larger than expected, implying that these SNPs are likely under selection. So one thing to keep in mind, though, is that selection is actually really complex, and it can act at different stages of the life cycle. So for a diploid sexually reproducing organism, you can get differential survival from zygotes to adults or viability selection. Genotypes may influence whether or not you breed, or sexual selection. You could have different numbers of offspring or eggs, fecundity selection, and there may be segregation distortion in the population or comedic selection. And so Christensen and Friedeberg in 1973 actually came up with this really elegant series of hypothesis tests for testing selection at these different stages of the life cycle called selection component analysis. If you saw John Kelly's talk, this was the same framework that he was talking about this morning. And so the original framework was designed for random mother-offspring pairs, but because we have really exhaustive sampling of our population, what we've done is basically adapt the statistical framework to take advantage of the fact that we know who dad is, we know who mom is, and we know all of the kids. So let's start with comedic selection. So in comedic selection, the null hypothesis is a heterozygote parent will transmit both alleles, both big A and little a, in equal frequency to their offspring. And so the way we're looking for this in our population is we basically take all of the trios for which one of the parents is heterozygote, and then we count up the number of offspring, we count up the genotype composition of these offspring. And then using these numbers, you can calculate the likelihood of observing these different genotype counts given the probability that the male transmits the big A allele PM or the probability the female transmits the big A allele PM. And so, ooh, that came out really bad. Anyway, so here's the likelihood surface. So PF is on the x-axis, PM is on the y-axis. And so for most alleles, you can't really see this, but you would expect to see that a really high likelihood around 0.5, so equal transmission of big A. We do see a couple SNPs where, for example, here it seems like the male is transmitting the big A allele about 90% of the time, and we simply use a likelihood ratio test to test for SNPs that deviate significantly from 0.5. Here's a Manhattan plot, and basically we found no evidence of comedic selection in females, but we found 4 different SNPs that showed really evidence of strong distortion in males. So that's comedic selection. The other 3, viability selection, sexual selection, and fecundity selection, we're using a similar approach here, and so I'm going to talk about them all together. So we do this in 2 separate ways. The first is just simple kind of chi-squared Fisher's exact test, where we're looking for genotype differences among different groups of individuals, so birds that survive versus those that don't, or birds that breed versus those that don't. However, we also have a lot of environmental data, and we know that environmental correlates may affect how well you survive or reproduce, or how many eggs you lay. And so we also take a mixed model approach, where we're modeling either survival, breeding status, or clutch size. We fit a number of random effects, such as an individual, year, natal nest, parent identity. And then we fit a whole host of possible environmental effects. This is the beauty and curse of having a lot of data, because you want to try everything. And so we consider different attributes of the individual that might be important, such as age, or inbreeding coefficient, immigrant status. We look at attributes of the territory in which a bird was born. So number of helpers we know can affect survival, the territory size, habitat composition, the age of its parents. And then we also fit a number of effects of a given year. So breeding density, how much rain there was, how dry it was, temperature, acorn abundance, etc. So basically what we do is we use model selection, find the best model for each of the phenotypes given these different traits, and then we add the genotype of each SNP one by one to test whether or not the SNP, after you control for all of these possible non-genetic confounding factors, you see an effect of the SNP still. So in the interest of time, I'm just going to talk about results for viability selection. So we looked at viability selections at a number of different life stages, so survival from day 11 to day 30, day 30 to day 90, which is when they reach nutritional independence, day 90 to about a year, and then survival to breed. And so when we first ran the genotype-only test, we only found significant SNPs in the early life stages and in females, so about 7 SNPs were significantly correlated with, significantly associated with survival from day 11 to day 30, 14 SNPs from day 30 to day 90. We then added in environmental variables and found that nestling weight is really important early on. Survival from day 30 to day 90 is associated with rainfall, and also 7 SNPs after you account for rainfall. Acorn abundance explains overwinter survival, which makes sense, and the natal territory and juvenile weight explains survival later on. All right, so I didn't talk about sexual selection and fecundity selection, but we're still working on those models now. What I've shown today is that we use gene dropping simulations to identify 67 SNPs under net selection across in our population from 1990 to 2013. We then looked at selection components analysis, and we found evidence for 4 SNPs under comedic selection and 21 SNPs under viability selection. These SNPs are different, and so one of our hopes in using selection component analysis is trying to understand whether or not there's evidence for antagonistic selection operating in our population. And also I hope that this makes it clear that by applying genomic tools in long-term demographic populations is a really powerful approach to studying short-term evolution. I'd like to thank, first of all, none of the work I could have done would be possible without all the interns and students who worked at Archbold and collected all the data through the years. I'd like to thank my collaborators, the various mentors I've had, and my funding sources, and I thank you for your attention. Right, so the offspring are genotyped at day 11, and we were worried about confounding with early viability selection, and so the only nest that we have